So I am recording to the cloud now, and we're almost done. It's last week. Only 3 more Zoom meetings left, including today. And so without further ado, I'm going to start talking about nerve structure. So let me call up a whiteboard. And when we talk about nerve structure and classification, this is really easy stuff. So it shouldn't take you long. If you've already read it, I'm sure you have a grasp of this. If you haven't, that's okay, because it's not difficult at all. First, I'm just going to write up here on my board nerve structure, and there's a quick, easy classification that we use to do that. So cranial nerves, first of all, in nerve structure, all nerves, I'll define what a nerve is in a minute, but I'm just going to write all nerves in general. They arise from the brain or spinal cord. That's where they are sort of so that's where their main connection is, all nerves in the human body, whether they're afferent or efferent neurons. That's their connection, that's their base. So what we mean by a nerve, and some students do confuse this, a nerve is different than a neuron. A neuron is a single cell. A nerve is made up of many, many axons or fibers. So a nerve is sort of defined as, I'm just going to write a bundle of axons, and sometimes fibers is used instead of the word axons when we describe nerves. And some are myelinated and some aren't within the nerve itself. So some are unmyelinated as well. And it's all just bundled up in connective tissue. And the good thing for us right now is we already learned how a muscle, an entire muscle is configured or structured, and a nerve is structured in almost the same way. We use the same language. So endoneurium is the delicate connective tissue that wraps around a single axon. So just like endomysium was the delicate connective tissue that wrapped around a muscle fiber, endoneurium is it's basically the same connective tissue. It's delicate, thin connective tissue that wraps around a single axon. So I'm just going to write around each axon. All this language is the same. And then perineurium, we just use the word neurium, the suffix neurium, instead of mysium, to know that we're talking about a neuron. Perineurium is coarser connective tissue that bundles up a bunch of axons. So this is around many axons that are already wrapped in endoneurium. And guess what? Forms bundles called fascicles, just like in a muscle. So we're just making bundles and bundles of things. And finally, epineurium, instead of epimysium in a muscle, epineurium is the coarsest connective tissue. So this is thin and delicate, this is coarser, this is the coarsest, thickest, coarsest connective tissue. And this encloses several fascicles to form the actual nerve. And there you have it. Now there's other things within a nerve besides just wrapped axons. So other tissue that's found within a nerve and structures, well, we know that myelin's going to be in there. Actually, myelin takes up probably the bulk of the volume of a nerve when the axons are myelinated. So we have myelin in there, of course. Also within a nerve, there is plenty of connective tissue. So I'm just going to write CT. There are blood vessels and lymph vessels, also found in that in the epineurium, in that wrapped nerve, are all of these tissues and structures. So that basically makes up the structural the structure of a nerve. Then there is a functional classification that we give nerves, which you'll also find quite simple and easy because you know all the language now. So the functional classifications of this business are just, we have, like I said, you know this language now. Motor nerves, you know those are going to be efferent, EFF, efferent nerves that carry impulses away from the central nervous system. And then there's going to be sensory nerves, and you know that word is associated with afferent. So sensory nerves are just bundles of fibers of axons that carry impulses toward the central nervous system. And then finally there are mixed nerves, which have a combination of both 1 and 2. I'm just going to write combo, C-O-M-B-O, of number 1 and 2. So it's got both types of fibers in it, both motor neurons, axons, and sensory neurons or axons. So I always tell students a good way to remember a mixed nerve is this is like a two-way highway. That just means within the nerve itself, we've got, so if this is a nerve that's running through our body, either through our appendages in the peripheral nervous system or in the central nervous system, we've got bundled axons transporting information this way as well as this way. So nerves are rarely just all motor or all sensory. That's not typical, that's unusual. These are the most common. Just like when you drive around in your car, especially in the United States, one-way streets, unless you're in Philly or in Center City or something like that, one-way streets are quite rare these days. Two-way streets are much more common, and that's the same with the nerves in our bodies. So I'm just going to give you a quick review in language. So the fibers, let's go back to this one and let's just layer on top of this. So we say the fibers within a nerve, within a mixed nerve, I should say. That's supposed to be an X. Are things like somatic, afferent. I'm going to expect you to know this language on the quiz. So somatic means external world, external environment. Afferent gives us the directional pathway toward the central nervous system. So these would be things, sensory nerves that are sensing the external environment. There's also visceral afferent fibers. So if we just dissect this term, visceral means from the inside of the body, internal organs. Afferent is the directional pathway toward the central nervous system. We also have somatic efferent fibers. Just to review the language, somatic means external environment. Efferent moving directional pathway going impulses transmitting away from the central nervous system. So these would be to things like skeletal muscles. And finally, there are visceral afferent fibers. Sorry, efferent. We already did visceral afferent. These would be direction of transmission away from the central nervous system to things on the inside of our body. These are to skeletal muscles. These would be to organs and glands, smooth muscle, cardiac muscle, and glands. So this is just language of types of fibers within a mixed nerve. As a rule, now I'm going to talk about damage to a nerve. So let's write a new header. So when damage occurs, the ability of a nerve to heal depends really on its location, first of all. So as a rule, mature neurons do not divide. They are amitotic. That was one of the first things you said in the nervous system. So nervous tissue in general is not regenerative. However, depending on where the damage occurs, there is an ability for the nerve for some regeneration. So I'm just going to talk briefly about that. Axons of the peripheral nervous system may regenerate. And that's not because they're stem cells, they're constantly cranking out new cells. These cells are amitotic. There are no stem cells. Once we're born with our neurons, we basically have to live with them. So thank goodness they have longevity, they have long lifespan. But there is an ability of axons in the peripheral nervous system to do some regeneration. And that's if the damage or injury typically is far away from or distal to the cell body. So I'm going to put is distal, and in parentheses, far from the cell body. It also depends on the degree of damage, the degree of injury. So really quickly, if this is an axon of let's say here's the cell body, and if this is an axon of a nerve, I'm going to draw it bigger. If we just crush this nerve cell a little bit with pressure, yes, there's some damage to it, but its chance of some regeneration is okay, is quite good, rather than if there's a complete sever in the tissue. If we cut this axon, the chances of regeneration are not nearly as good for obvious reasons. So before I go to another page, this is worth saying. Axons of the central nervous system typically cannot regenerate. So we say, you've probably heard the expression that once you lose brain cells, you're not getting them back. And that's pretty much true. That's true. Cells in the central nervous system, once they're damaged, they're pretty much gone for good. And the difference between the two, the ability of peripheral nervous system neurons to regenerate as opposed to central nervous system, is based on this. I'm going to just jump to a new whiteboard. And I'm just going to write difference of the ability of the peripheral nervous system to regenerate and the central nervous system cells not to generate is due to Schwann cells versus oligodendrocytes. Now, we already talked about this. Schwann cells are the cells that myelinate axons, fibers, in the peripheral nervous system. That's what's going to give them the ability to regenerate. Oligodendrocytes, they myelinate the axons or fibers in the central nervous system, and they are what prevents, one of the major causes that prevents the central nervous system fibers from regeneration. So I'm just going to write Schwann cells. And they help. I'm going to talk about that next. PNS regeneration. They not only myelinate, but they help with regeneration, as opposed to these oligodendrocytes. These actively suppress regeneration. I'm going to put CNS regeneration. That's the main difference in their abilities to do any sort of healing or regeneration. And in particular, oligodendrocytes, I'll say one more thing about this is just a myelinating material in the central nervous system. It kind of oozes around and covers the fibers, and it helps with insulation and electrical transmission, speeding of transmission. But these oligodendrocytes are also covered, that's a W, they're covered with growth inhibiting proteins. Also astrocytes in the CNS. Remember that neuroglial class of cells, astrocytes, those star-shaped cells in the central nervous system, they're the most common. And they perform so many functions structurally and all sorts of things, but these, when there's damage in the central nervous system, astrocytes quickly form scar tissue, which doesn't help regeneration of new neurons. So if asked, why is regeneration so poor or non-existent in the central nervous system? It's because of this. Oligodendrocytes actively suppress regeneration because they're covered with growth inhibiting proteins, and astrocytes in the central nervous system quickly form scar tissue when any damage occurs. All right, I'm going to label over to a new board here and just talk about, now I'm going to talk about axon regeneration in the peripheral nervous system. So we know that those Schwann cells are a key player in this. I'm going to refer you to figure 13.31. So I'm going to write that down because it describes this whole process. And that's in your textbook. The regrowth is never exactly the same. So when this growth occurs, and there's steps on table 13.31 in your textbook, and I'm not going to redraw the entire diagram or figure, but there are 1, 2, 3, 4 steps. I'm going to mention them. When there is damage to a fiber or an axon, the axon function is interrupted, and hopefully this is distal to the cell body. The main player is going to be Schwann cells in a myelinated nerve cell, and ultimately those Schwann cells and macrophages play a key role as well. So let's just say we have a myelinated, here's an axon, and here's myelin around that axon, wrapped around it. If we get damage in this area of the axon, the key player are going to be these Schwann cells out here. They ignite macrophages, which are immune cells, to clean up the debris and sort of clean out the area. And then in turn, macrophages also help stimulate the Schwann cells to secrete something called nerve growth factor. And what they do, the Schwann cells, when they secrete this chemical called nerve growth factor, is they create this bridge, sort of like in a broken bone, that callus that forms. These Schwann cells are going to form this protective tube in this area, and they secrete a chemical called nerve growth factor that stimulates this neural tissue to start growing and re-bridge this gap here. I'm simplifying this process, and actually so does your book. And when this neural tissue starts to grow back, we do get regrowth of the axon. We get more myelination out here to protect this area, insulate it, and help with transmission. But at the end of all of this process, the regrowth is never exactly the same, unlike a bone. It's not quite that neat. And this transmission pathway of, let's say this axon is firing an impulse, oftentimes through physical therapy and other activities, we need to sort of retrain this transmission pathway at this breakage area for it to fire to the correct effector muscle or muscle. So in severe nerve damage, if you lose, say, the function of the effector organ, let's just say skeletal muscle at this end, due to the nerve damage, we can get regrowth and regeneration here, but oftentimes this neural pathway needs to be retrained over and over and over again so that the muscle can work as it should originally. I'm going to highlight, since you guys showed up for this lecture, I am going to highlight back in step one of this regenerative process, there's something called Wallerian degeneration. That's nothing magical, but if you see this crop up on the quiz, that's just a term that's used for the degradation of the tissue here after damage. So it just represents degeneration of the cut axon. And this tissue distal to the, if this is the cell body, the soma, the tissue distal, I'm going to use a different color because it's getting messy. The axon tissue distal to the damage area starts to degenerate, and that's what Wallerian degeneration stands for, just the degeneration of the damaged tissue. All right, let's move on now to chapter 14. Sorry, 13.7. I need to just mention that really quickly. So let's layer on here. So 13.7 refers to cranial nerve. I'm not going to go through, I'm going to tell you in this lecture what I will expect you to be able to access or know during the quiz, okay? So 13.7 is all about cranial nerves. There's 12 pairs of nerves in the brain. So 12 pairs of cranial nerves. That means we have 24 total cranial nerves. They are numbered using the Roman numeral system. So when you see them in a textbook, or if I write a specific number of a cranial nerve, it's going to be in Roman numerals, not in Arabic numerals. So I'm going to write this now, page 475 in the text. That just gives a short list of all 12 and their basic functions. That's what you should focus on in your studies, okay? Then the rest of this section goes on and details each of those 12 cranial nerves. We just don't have the time to go in and discuss each little tiny detail. I'm going to be focusing on the list on page 475 when I ask you a few questions on the text. This is worth noting and highlighting. Only cranial nerve number 10 which is called the vagus nerve. This is the only cranial nerve that extends further than the head and neck. So 11 of the 12 cranial nerves only do their business in the cranium, in the head and the neck. Only the vagus nerve, number 10, extends. It extends down all the way down into the abdominal cavity. So that's worth noting because it's sort of special. And the numbering of 1 through 12 is done in a rostral to caudal direction. That just means think of from the front to the back. This word rostral just means snout. And caudal means tail. So I'm just going to put in parentheses anterior, from the sort of the front of the head or the front of the brain, to posterior. By posterior, that I mean not only the back of the brain, but also down into the brain stem. Okay, and so when you look at an inferior view, which is shown on page 474 of your book, you'll see a general categorization of numbering from top to bottom. That's really going from anterior to posterior or front to back. So make sure you focus on this overview of this list on page 475. Now I need to get into the autonomic nervous system before we call it a day. So that's a new chapter. We're going into chapter 14. And I'm going to talk about the autonomic nervous system. And we know that it regulates internal body business or internal body systems. Specifically, it innervates smooth muscle, cardiac muscle, and glands. And these are called the effectors. That should not be a totally foreign word to you. So the effector organs or the effectors are smooth muscle, cardiac muscle, and glands. You know what? Before I do this, before I go any further, just to remind you, I know we did this a long time ago, but just to give you context, the nervous system does this. We have sensory receptors that send impulses to the central nervous system, where those impulses are processed, and then a command is sent out from the brain and spinal cord. So this is sensory afferent. This would be motor efferent. And in this pathway, we have a somatic division that goes to skeletal muscles and an autonomic division. The word autonomic, by the way, just means self-governing. I tell students, try to just think autonomic, automatic. And there's two subdivisions of this, parasympathetic and sympathetic. And that is what this discussion is about, this business right here. So we're talking about a motor efferent pathway from the central nervous system that innervates smooth muscle, cardiac muscle, and glands. And I will mention the two subdivisions today. Most of that is going to be lectured tomorrow. But let's just talk about the autonomic nervous system now, okay? It's the motor subdivision of the nervous system that we call involuntary or general visceral system. So we already mentioned the effectors, which are smooth muscle, cardiac muscle, and glands. But I'm going to specifically talk about autonomic for the rest of this lecture. Autonomic, efferent. I'm going to use this word just to remind you the direction of flow. Pathways. And ganglia. Sorry, I just had to shut off the phone. So efferent pathways and ganglia, the autonomic nervous system uses a two-chain pathway, unlike the somatic division. So that if you understand the basics of this, the rest of the discussion will be easier to understand. So on that pathway, going from the central nervous system outward in the motor afferent pathway, if we go to the somatic division, which is skeletal muscle, that's a one-chain pathway. These motor neurons are super long, meaning the cell body of this multipolar neuron is up here in the central nervous system, and then its axon extends all the way to the skeletal muscle where we find the terminals. That's the somatic. The autonomic nervous system uses a two-chain pathway. So the autonomic pathway does this. Typically, the preganglionic fibers, the cell bodies are located in the central nervous system somewhere. And then they go to make a two-chain connection. So this is the autonomic. And then the termination is on the effector organ, which is smooth muscle, cardiac, and glands. The key is this pathway of the autonomic nervous system, it's a two-chain system. So there's a synapse here somewhere between the central nervous system and the effector organs. There's another neuron, there's a synapse here, and it always uses a two-chain pathway. The somatic division, which we are not really talking about right now, just so you know, the difference is it uses a one neuron, one chain pathway. So that's the first thing to get through to understanding this. Now, within this two-chain pathway, so I'm just going to write up here this two-chain pathway that I just pictured. The cell body of what we call the preganglionic nerve. Ganglion in this case is just going to mean synapse. Technically it means clusters of cell bodies in the peripheral nervous system, but for this discussion, try to think of the word ganglion as a synapse, a connection point. So the cell body of the preganglionic neuron is in the central nervous system, the brain or the spinal cord, okay? Its preganglionic axon This is just getting used to the language. The preganglionic axon synapses with a second motor neuron somewhere outside in the peripheral nervous system. So I'm just writing synapses with a second motor neuron, and we're going to call that the postganglionic neuron. Okay. Second bit of this is that the postganglionic neuron has a cell body in an autonomic ganglion outside of the central nervous system. This is just called an autonomic ganglion in the peripheral nervous system somewhere. That postganglionic axon then is what reaches to the effector. This is just fancy, complicated language to describe what I drew in this previous picture. I'm just describing in words this pathway. The cell body of the preganglionic neuron is found in the central nervous system, and it extends somewhere out into the peripheral nervous system where there is a synapse that we call an autonomic ganglion, and then a new neuron, the postganglionic neuron cell body is here, and its fibers extend all the way to the effector organ. So we've just, in word form, just described that pathway, that's all. I'm going to go back to this page now and highlight. This is all described in figure 14.2 in your textbook. That, I'm going to, sorry to keep flipping around like that, but figure 14.2 is just going to give you this is a simplified version that I've drawn here. Meaning, I first drew this somatic motor neuron, and then I drew this autonomic chain pathway, okay? And figure 14.2 doesn't give you an overall big picture like this, but it's a comparison chart of the similarities, which aren't that many, but mostly differences between the red and the yellow pathways. So I'm going to now give you some just characteristics of this yellow pathway. So that's what I was about to do on this blank board. So preganglionic fibers, and I hate to keep going back and forth, but just so you know, we're talking about preganglionic fibers. In this case, we're talking about these, starting here and going to this juncture. So this would be a preganglionic fiber, okay? Now I'm going to quickly go back to these fibers, and they're just axons is what we're talking about. These tend to be thin and very lightly myelinated. And then postganglionic fibers in this pathway, that's just the second part of the chain that goes all the way to the effector organ. These are even thinner. Sorry, I'm having a little bit of difficulty on this board this morning. These are thinner and completely unmyelinated. So that tells us something about them. So we know that, we go back real fast to my pathway picture. So what we just said is these preganglionic fibers are typically thin and lightly myelinated. And these postganglionic fibers are even thinner and completely unmyelinated. So that tells us that that entire pathway is going to be very slow, especially compared to these guys, which are typically heavily myelinated and super fast. All right, I'm going to go back to here. All somatic motor neurons. That was the red pathway on my picture. Release acetylcholine at their terminal end. So all somatic motor neurons release acetylcholine as neurotransmitter, and it's excitatory. And I'm going to write skeletal muscles. Jump back real fast. So all we said is this red pathway to skeletal muscles always uses acetylcholine when it innervates skeletal muscles, and it always excites them, causes them to depolarize and then contract. That's different from the autonomic nervous system. So I'm going to draw a break here and make statements about the autonomic nervous system. All preganglionic visceral motor neurons, I'll write autonomic to help you with the language, secrete, these are motor neurons, remember, that just means they're going away from the central nervous system. These secrete acetylcholine as well, but hold the thought. So I'm just going to write preganglionic visceral. Remember, this is the yellow pathway, so I'm going to use yellow to underline them. They use acetylcholine. All that statement is saying is this: in this pathway, right, this first neuron, this one, the preganglionic nerve fiber, it releases acetylcholine at this juncture. Now, all postganglionic visceral autonomic neurons, they secrete either, I'm going to underline either, acetylcholine, I'm going to write ACH for its abbreviation, or norepinephrine. And the result at the effector organ can be either excitatory or inhibitory. So I'm going to go back to my picture now. And we're talking about this second pathway. I'm going to use it. I'll get back to my yellow. So we're talking about this pathway. What's released here at the postganglionic terminus is either acetylcholine or norepinephrine. And the effects are not always excitatory like they are in this case. They're either excitatory or inhibitory. Depending on the receptor at the effector. Okay, so it gets the red pathway is more simplistic than the yellow pathway. It's several fold. It's a one neuron chain, and it always releases acetylcholine, and it's always excitatory. That is somatic skeletal muscles. The autonomic nervous system uses a two-chain system called pre- and post-ganglionic neurons. The preganglionic neuron always releases acetylcholine at this stage, at the ganglion, the synapse, and then the postganglionic fibers release either acetylcholine or norepinephrine, and the results of those neurotransmitter can be either excitatory or inhibitory depending on the receptor at the effector organ. All right, guys, that is, I know that this was a lot of stuff. Well, actually, it was not that much information, but it can get complicated with these autonomic effects. I'm going to highlight this before I leave. I'll talk more about this tomorrow, but there are two subdivisions of this autonomic system. So let me just go back, quickly highlight that. So in the autonomic, this is mostly what I'm going to talk about tomorrow. There is a parasympathetic division and sympathetic division. What I was going to say is that it's the sympathetic division that at the effector norepinephrine is used as neurotransmitter. And the parasympathetic acetylcholine is used as neurotransmitter. I'll talk more about these subdivisions at our next lecture, okay? So I'm going to call it good. I will stop recording and then give you a chance to,